Tonight we are going to do something unusual. We are going to follow a single question, one of the most extraordinary questions in all of science. From the smallest nervous system ever mapped to the largest one that has ever existed. And by the end, you will understand exactly how far we have come and exactly how far we have left to go. The question is simple. Can you copy a mind beat? Not simulate one. Not build one from scratch. Copy one. Take a living, conscious, thinking mind, yours, mine, anyone's, and reproduce it so faithfully that the copy wakes up and remembers breakfast. That is the question at the center of a quiet revolution happening in laboratories around the world right now. And the answer, the real, honest, scientific answer, is more complicated and more astonishing than you might expect. There is a television show called Pantheon. It aired on AMC in 2022. In it, a dying man's mind is scanned, destructively, irreversibly, and uploaded into a computer. He wakes up inside a digital environment. He remembers his wife. He remembers his daughter. He is, by every measure available to him, still himself. The show treats this as a near-future thriller. The technology is portrayed as dangerous, secret, and roughly 10 to 15 years away from the present day. Tonight we are going to walk through the real science behind that premise. Not to debunk it, not to hype it, just to ask, honestly, how much of this is fiction and how much of it is already happening. Beat. The answer will take us through worms, flies, flatworms, octopuses, the most detailed brain scan ever performed, and a question about identity that has no clean answer. We are going to move slowly. This is not a sprint. There is no quiz at the end, no test, no moment where you need to have memorized a fact. This is a walk through some of the most remarkable science that human beings have ever produced. And the only thing you need to do is follow along and let the ideas accumulate. They will build on each other. Each one will make the next one more astonishing. Let us start at the beginning, not the human brain, something far smaller. Breathe slowly. There is no urgency here. We have time. There is a worm. It is about 1 millimeter long. It lives in soil. It eats bacteria. It has no eyes, no ears, no lungs, no heart. It has exactly 302 neurons. Its name is C. No rhabditis elegans. Scientists call it C. elegans. And it is, quietly, one of the most important organisms in the history of neuroscience. In the early 1970s, a South African biologist named Sidney Brenner made a decision that would shape the rest of his career and change the trajectory of an entire field. He decided to map every single neuron in the nervous system of this worm. Every cell, every connection, every synapse. The complete wiring diagram of a living brain. This had never been done before, for any organism, at any scale. The very idea that you could create such a map, that you could look at a nervous system and say, here is every connection, and I have accounted for all of them, was itself a radical proposition. Consider the context. In the early 1970s, scientists understood that brains were made of neurons. They understood that neurons communicated through synapses. They had recording equipment that could listen to a single neuron firing. But the idea that you could map every connection in an entire nervous system, even a tiny one, was like suggesting you could count every grain of sand on a specific beach. Technically possible, practically insane. Brenner chose C. elegans for a reason. Among all the animals he could have studied, this worm had the smallest nervous system that still produced recognizable behavior. It moved, it fed, it responded to touch and temperature and chemical gradients. It mated, it laid eggs. All of this from 302 neurons. If you were going to attempt the impossible, a complete map of a nervous system, this was the organism where the impossible was closest to merely very difficult. The method was brutal and painstaking. You take the worm, you embed it in resin. You slice it into sections thinner than a wavelength of light, 30 to 50 nanometers each. A nanometer is 1/1000th of a meter. A human hair is roughly 80,000 nanometers wide. These slices are thinner than visible light itself. You place each section under an electron microscope. You photograph it. Then you move to the next section, and the next, and the next. Thousands of sections, each one a single frozen plane through the body of this creature. Then you sit down with the photographs and you trace, by hand, every neuron, from section to section, through the entire body. You note where one neuron touches another. You identify the synapses, the junctions where signals pass from one cell to the next. You follow each thread of nervous tissue through the stack of images, matching shapes from one slice to the next, building a three-dimensional picture, one painstaking layer at a time. Think about what this actually involves. You are looking at a black and white photograph of a cross-section of an organism smaller than the head of a pin. The photograph shows a tangle of membranes and organelles. Somewhere in that tangle are the cross-sections of neurons, circles and ovals and irregular shapes that represent the axons and dendrites cut through at this particular angle. You need to identify each one. You need to label it. And then you need to find the same neuron in the next photograph, and the one after that, and the one after that, tracking each cell through hundreds or thousands of slices as it branches and bends and connects to other cells. There is no automation. There is no AI. There is a person, a microscope, a stack of photographs, and a pencil. 15 years. That is how long it took. Think about that for a moment. 15 years of tracing neurons through electron micrographs, more than half of a typical scientific career. The kind of commitment that only makes sense if you believe, deeply and persistently, that the result will matter long after you are finished. People joined the project and left it. Graduate students became professors. Children were born and started school and graduated. The Cold War continued and ended. Computers went from filling rooms to sitting on desks. Punk rock happened. Disco happened. The Berlin Wall fell. And through all of it, someone was sitting at a bench, tracing neurons through a stack of images, one slice at a time. This was not glamorous work. It was not the kind of science that makes headlines or wins grants easily. It was the kind of science that requires a particular temperament, a willingness to do something tedious and repetitive for years, sustained by the conviction that the result would justify the cost. The paper was published in 1986. White, Southgate, Thomson, and Brenner. The structure of the nervous system of the nematode Caenorhabditis elegans. It filled an entire issue of the Philosophical Transactions of the Royal Society. 340 pages, 302 neurons, roughly 7,000 to 8,000 synaptic connections. Every single one accounted for. Sidney Brenner would later share the Nobel Prize in 2002. Partly for this work, partly because the map he built turned out to be the foundation for something no one fully anticipated at the time. What Brenner and his colleagues had created was not just a picture, it was a connectome, a word that would not be coined until 2005, when Olaf Sporns, Giulio Tononi, and Rolf Cotter published a paper proposing the term. But the concept was there from the beginning. The complete wiring diagram of a nervous system. A map not of what the brain does, but of what the brain is. Every neuron, every connection, the entire architecture laid bare a blueprint, a schematic, a parts list, and assembly diagram for a living mind. And a connectome is a strange and powerful thing, because a connectome is not a description of behavior. It is not a list of the things a worm does. It is the structure from which behavior emerges. This distinction matters enormously for everything that follows. So let us make it concrete. Let us take a moment here. The idea of a connectome is deceptively simple, and it is worth sitting with before we move on. When you look at a road map, you see where roads go. You see intersections, highways, back streets, dead ends. The map tells you the structure of the transportation system, but the map does not tell you that most people drive to work between 7 and 9 in the morning, or that the highway gets jammed every Friday at 4, or that the back road behind the school is only busy during drop-off and pickup. The map shows what is possible. The traffic shows what actually happens. A connectome is a map. It shows you the roads. The question, the one that drives everything we are about to discuss, is whether the roads are enough, whether the structure alone can explain the traffic, whether the wiring diagram by itself can produce a mind. The worm gave us the first chance to test this. In 2014, a group of researchers working on an open-source project called OpenWorm built something remarkable. They took the connectome, all 302 neurons, all 7 to 8,000 connections, and they simulated it, not on paper, in software, a computational model of every neuron in the worm's body, connected exactly as they are connected in the living animal, running according to the mathematical equations that describe how real neurons fire. The simulation included not just the neurons but 95 muscle cells. It used a 3-dimensional physics engine to model the worm's body moving through space. The equations governing each neuron were Hodgkin-Huxley models, the same mathematical framework that won Alan Hodgkin and Andrew Huxley their own Nobel Prize in 1960. 3 for describing how nerve impulses propagate. This was a digital worm, not an approximation, not a cartoon, a simulation built from the actual measured architecture of a real animal's nervous system. And then they pressed play. The digital worm moved. It exhibited forward locomotion, the characteristic sinusoidal wave that C. elegans produces as it swims through its environment. Picture a snake moving across sand. That side-to-side undulation. Each curve propagating backward along the body, pushing the animal forward. That is what the simulation produced. Not because anyone programmed undulation, because the connectome, when activated, naturally generates alternating patterns of muscle contraction on opposite sides of the body. And those alternating contractions produce a wave. The movement was not perfect. The worm's turning behavior was less accurate than its forward motion. Some of the subtler behaviors, responses to specific chemicals, for instance, did not appear in the simulation. But the basic motor pattern was there, emerging from nothing but the connectome and the physics of a simulated body. This was remarkable. But the most striking demonstration came from outside the OpenWorm project. In 2014, a researcher named Timothy Busby presented something at the Solid conference that sounds like it should not be real. He took the connectome, the wiring diagram from Brenner's paper, and he loaded it into a small robot built from Lego bricks. The robot had a sonar sensor, where the worm's nose would be. It had motors on each side where the worm's body muscles would be. And the control system, the thing that decided what the robot would do, was not a program. It was not code. It was the connectome. 302 simulated neurons, connected exactly as they are connected in the living worm, running in real time on a simple processor. No behavior was programmed. No instructions were written. No one told the robot to move forward, or to avoid obstacles, or to respond to touch. The connectome was loaded. The robot was switched on, and they watched. The robot moved forward. When it encountered an obstacle, it turned away. When something touched its front sensor, it reversed direction. When something touched its rear, it moved forward again. These are the same behaviors the living worm exhibits in its soil environment, and none of them were coded. They emerged spontaneously, naturally, inevitably, from the structure of the connections alone. The connectome was the behavior. Let that settle for a moment. 302 neurons, connected in a specific pattern, and the pattern itself produces movement, produces avoidance, produces something that looks remarkably like decision-making, not because anyone designed the behavior, not because someone wrote rules for how to avoid walls. The wiring is the logic. the map is the mind. At least for this worm, at least at this scale. There is a philosophical implication here that is easy to miss in the excitement of a robot that moves like a worm. The Lego robot was not made of biological tissue. It was not running in a chemical environment. It did not have neurotransmitters or ion channels, or any of the molecular machinery that a real nervous system depends on. It had a microprocessor running a mathematical approximation of neural activity, connected in the same pattern as the worm's neurons, and it produced the same behavior. This suggests something profound. The behavior does not require the specific material. It does not require biology. It requires the pattern, the information, the map. Run that map on any substrate, biological neurons, silicon chips, Lego bricks, and the same behaviors emerge. Or at least they do for 302 neurons. The question is whether this principle holds as you scale up, whether the principle that works for a creature simpler than a thermostat still works for a creature that can learn, that can navigate, that can dream, whether the map remains the mind as the map grows from a sketch on a napkin to a blueprint the size of a city. That question hangs over everything that follows. Hold it gently. We are going to carry it up the ladder with us. Now hold that thought, because the next organism up the ladder tells us something even more remarkable. For 38 years after Brenner's paper, the C. elegans connectome stood alone, the only complete wiring diagram of any brain in existence. 302 neurons, a monument to patience and microscopy. No one completed another one. Not because no one tried, because the next organisms up the ladder were so much more complex that the methods available simply could not handle them. Mapping 302 neurons took 15 years by hand. Mapping thousands, or hundreds of thousands, would take centuries with the same approach. What changed was technology. Two things, in particular. The first was automated electron microscopy, machines that could slice and image tissue continuously, without a human operator switching samples and adjusting focus between every frame. The process that Brenner's team did manually, slice, photograph, move to the next, could now be done by robots running around the clock. The second was artificial intelligence, neural network algorithms that could look at an electron micrograph and identify neurons automatically. Not perfectly, not without errors, but well enough that a human proofreader could correct the AI's mistakes in a fraction of the time it would take to trace every neuron from scratch. These two advances, faster imaging and AI-assisted tracing, compressed what would have been centuries of work into years. And in October 2024, a consortium of researchers published the complete connectome of an adult fruit fly. The organism is Drosophila melanogaster, the common fruit fly. You have seen them in your kitchen. They are 3 millimeters long. They live for about 30 days, and they have roughly 9,000 neurons, connected by more than 50 million synapses. The scale difference from C. elegans is staggering. 302 neurons to 139,000. 8,000 synapses to 50 million. 460 times more neurons. 6,000 times more connections. The project was called FlyWire. It was a collaboration between the MRC Laboratory of Molecular Biology in Cambridge, Princeton University, the University of Vermont, and dozens of other institutions around the world. The raw data came from a single female fruit fly. Her brain was fixed, embedded in resin, and sliced into roughly 7,000 sections. Each section was imaged under an electron microscope at a resolution sufficient to see individual synapses. The resulting image stack was enormous. Tens of terabytes of data representing every cubic nanometer of the fly's brain. Then the AI went to work. Convolutional neural networks, the same class of algorithms used in image recognition, segmented the images into individual neurons. The algorithms traced each neuron's branches through the three-dimensional stack, identifying where axons connected to dendrites, where synapses formed, where one neuron's output became another neuron's input. But the AI was not perfect. It made mistakes. It confused two neurons that ran close together. It lost track of thin axons that faded in and out of the image stack. It misidentified synapses, or missed them entirely. This is where the human proofreaders came in. Hundreds of researchers, many of them volunteers, sat at computer screens and corrected the AI's work. They examined flagged regions where the AI's confidence was low. They split neurons that had been incorrectly merged. They joined fragments that belonged to the same cell. They verified synapses, one by one. This proofreading effort took years. It was, in many ways, a modern echo of what Brenner's team did by hand. Except now, the humans were correcting a machine's first draft, rather than starting from blank photographs. The paper was published in Nature in September 2024. 9 companion papers were published alongside it, each analyzing different aspects of the connectome: the sensory systems, the motor circuits, the neuromodulatory pathways, the developmental patterns. Together, these papers represent one of the most comprehensive descriptions of any biological system ever produced. It is the second complete connectome in history, and it is, by any reasonable measure, the most complex biological structure that has ever been fully described. Let that register. In 1986, the pinnacle of connectomics was 302 neurons in a worm. In 2024, just 38 years later, it is 139,000 neurons in an insect. The scale increased by a factor of 460. The time was roughly the same as a human generation. Imagine what the next generation might map. But here is what makes the fruit fly connectome genuinely astonishing. Far more than a simple scaling up of the worm experiment. Shortly after the FlyWire connectome was published, a company called Eon Systems did something that echoes what Busby's did with the Lego robot. But at a completely different level of complexity, they took the FlyWire connectome, all 139,000 neurons, all 50 million connections, and they built a computational model of it, a simulation of the entire fruit fly brain. The approach was conceptually identical to what OpenWorm had done with the worm. Take the connectome, assign each neuron a mathematical model that describes how it fires in response to input. Connect the neurons exactly as they are connected in the real brain. Run the simulation, see what happens. But the scale was different by 3 orders of magnitude, and the test was different too. They compared the motor output of their simulated brain, the patterns of muscle activation it produced in response to sensory input, to the motor output of living flies performing the same tasks: walking, turning, responding to visual stimuli, grooming, the kinds of everyday behaviors that a fruit fly performs thousands of times in its short life. The accuracy was 95%. Let that number register. A simulation built entirely from the structural connectome. No learning algorithms, no training data, no neural network optimization, no parameter tuning against behavioral observations, reproduced 95% of the real animal's motor behavior. The wiring diagram alone, run as a computational model, produced a virtual fly that moved almost identically to a real one. Think about what this means in terms of our roadmap analogy. Someone handed you a map of every road in a city. No traffic data, no usage statistics, no information about time of day or rush hour patterns, just the roads. And from the roads alone, you predicted the traffic with 95% accuracy. That should not be possible. Traffic depends on thousands of factors beyond road layout: population density, work schedules, weather, construction, accidents, human preferences. And yet, if you somehow managed 95% accuracy from road layout alone, you would have to seriously reconsider your assumption that the layout was not the main thing. That is exactly where connectome science stands right now. Remember what happened with the worm: 302 neurons loaded into a Lego robot, and behavior emerged from structure. Now the same principle, scaled up by a factor of 460, and the result is not just vaguely similar to real behavior. It is 95% accurate. This is profound, and it is important to understand exactly what it does and does not prove. It proves that connectome structure is sufficient to reproduce motor behavior with remarkable fidelity, even in a brain complex enough to coordinate flight, navigation, courtship, and foraging. A fruit fly is not a simple organism. It can learn, it can remember, it can navigate complex three-dimensional environments. It performs elaborate courtship dances. It adjusts its behavior based on past experience. All of this emerges from 139,000 neurons. It does not prove that the fly is conscious. It does not prove that the simulation experiences anything. It does not prove that a human connectome would produce a human mind when simulated. But it dramatically narrows the space of possible objections. If you claimed, before FlyWire, that structure alone could never produce realistic behavior, that the connectome was merely a skeleton, and the real brain lived in its chemistry, its timing, its dynamic flux, you now have to explain 95% accuracy. The 5% gap is interesting too. What lives in that gap? Is it noise, measurement error, slight differences between individual flies? Or is it signal? The contribution of neuromodulation, synaptic strength, timing dynamics, all the things that a static connectome cannot capture. We do not know yet, but 5% is a very small place for all of the soul to hide. The worm was a hint. The fly is evidence, not proof evidence. The distinction matters. Science does not deal in proof outside of mathematics. It deals in evidence, in probability, in the accumulation of results that make one hypothesis more plausible and another less so. The fly connectome does not prove that the map is the mind, but it makes the hypothesis that structure is sufficient far more credible than it was before, and it puts the burden of explanation on those who claim otherwise. If you believe the mind is more than its wiring, you now have to explain why the wiring alone gets you 95% of the way there. Breathe slowly. We are climbing a ladder, and each rung changes what we thought we knew. Between the worm and the fly, other organisms have been partially mapped. Each one taught us something the last one could not. The zebrafish larva has roughly 100,000 neurons. Its brain has not been completely mapped yet. A full connectome remains in progress, but the zebrafish offers something no other model organism can. It is transparent. A larval zebrafish is nearly see-through. If you hold one up to a light, you can see its organs, its blood vessels, its brain. And this transparency is not just convenient. It enables a form of neuroscience that is impossible in any opaque organism. Picture this. You are looking through the eyepiece of a specialized microscope. Below you is a larval zebrafish, barely 4 millimeters long, suspended in a droplet of water on a glass slide. The animal is alive. Its heart is beating, its eyes are moving, and you can see through the transparent skin and skull the faint gray mass of its brain. Now imagine that every neuron in that brain is engineered to glow green when it fires. That is not science fiction. That is calcium imaging. The technique works like this. Neurons communicate by firing, generating brief electrical impulses that travel along their length and trigger the release of neurotransmitter at the synapse. Says, when a neuron fires, calcium ions flood into the cell. This is a fundamental part of the signaling process. Calcium enters, triggers neurotransmitter release, and then is pumped back out as the neuron resets. Scientists have engineered proteins that glow when they bind to calcium. These proteins, called genetically encoded calcium indicators, with names like GCAMP, can be inserted into the genome of the zebrafish so that every neuron in the animal's brain produces the glowing protein. The most commonly used variant, GCAMP6, was developed by Lauren Luja and colleagues at the Howard Hughes Medical Institute's Genelia Research Campus. In 2013, it glows brightly within milliseconds of a neuron firing and fades as the calcium is pumped out. The brightness is proportional to the amount of calcium, and therefore to the intensity of the neural activity. A neuron firing once produces a brief, dim flash. A neuron firing in a rapid burst produces a sustained, bright glow. When a neuron fires, calcium floods in, the protein binds it, and the neuron lights up. When the neuron stops firing, the calcium is pumped out, and the glow fades. Because the zebrafish is transparent, you can see this glow from outside the animal with a specialized microscope, typically a light sheet microscope that illuminates a single thin plane of tissue at a time. You can watch individual neurons firing in a living, behaving animal in real time, every neuron in the brain simultaneously. Let us be specific about what this microscope does. A light sheet microscope sends a thin blade of laser light through the brain from the side, illuminating only a single two-dimensional plane, perhaps 10 micrometers thick. A camera positioned above the fish captures an image of that illuminated plane. Then the light sheet moves to the next plane, and the next, sweeping through the entire brain in a fraction of a second, capturing each plane as a separate image. Stack the images together, and you have a three-dimensional snapshot of every neuron that was active at that instant. Do this continuously, and you have a movie, a four-dimensional recording of the entire brain's activity over time. The first team to achieve whole brain imaging in a living zebrafish was led by Misha Ahrens and Philip Keller at Genelia Research Campus. In 2013, they published a landmark paper in Nature Methods, reporting that they had recorded the activity of roughly 80% of the neurons in a larva. Our zebrafish brain, approximately 100,000 neurons, simultaneously, at a rate of 1 volume per 1.3 seconds. That is an image of the entire brain. Every neuron captured faster than a human heartbeat. Researchers have done exactly this. They have recorded the activity of every neuron in a larval zebrafish brain, tens of thousands of neurons, while the animal swims, hunts, and responds to its environment. No other vertebrate brain has been observed this completely, while still alive and functioning. There is no rush here. Sit with this for a moment. We are talking about watching an entire brain think, not inferring its activity from electrodes, not reconstructing it from blood flow signals, as functional MRI does, actually watching each neuron light up and go dark in real time. In a living animal that is itself watching, deciding, and acting. Nothing like this has ever been possible in any other vertebrate. This matters for a specific reason. The connectome tells you where the roads are. Calcium imaging tells you where the traffic is. The zebrafish is the first animal where we can see both at the same time. The structure and the activity together in a living brain. And what the zebrafish has shown us is both confirming and humbling. The activity patterns are not random. They are not uniformly distributed across all neurons. Specific circuits light up during specific behaviors, exactly as the connectome would predict. When the fish turns left, the neurons that connect to left, turning muscles activate in a crisp, sequential cascade. First, the reticulospinal neurons in the hindbrain, then the motor neurons in the spinal cord. Then the muscles contract. The circuit fires in order, like dominoes falling along a path that the connectome drew. When the fish sees a potential meal, a tiny paramecium drifting past, the visual processing circuits light up in sequence. Detection in the optic tectum, a midbrain structure that acts as the zebrafish's primary visual processing center. Then, tracking, as the tectum locks onto the moving object. Then, an approach signal transmitted to the motor centers. Then, the strike, a rapid, coordinated lunge that captures the prey in less than 1/10 of a second. You can watch this entire sequence unfold in the calcium imaging data, each step activating a distinct population of neurons. each population connected to the next in exactly the way the emerging connectome predicts. But the activity also reveals something the connectome alone cannot. The same circuits produce different patterns of activity depending on the animal's internal state. A hungry zebrafish responds to prey differently than a satiated one. Even though the connectome has not changed, the circuit is the same. The activity on the circuit is different. Something beyond the wiring, something about the chemistry, the modulatory state. The history of recent activity is shaping the behavior. In the Aaron's lab experiments, this was visible in striking detail. Zebrafish that had been deprived of food for several hours showed dramatically increased activity in the prey. Detection circuits of the optic tectum, the same neurons, the same connections, but the gain had been turned up. The threshold for triggering a hunting strike was lower. The circuits were more sensitive, more responsive, more eager. And this heightened responsiveness was not localized to one circuit. It spread across the brain, altering the activity patterns of thousands of neurons simultaneously. The roads had not changed, but the weather had, the internal chemical weather of hunger, and the traffic pattern was transformed. The zebrafish is telling us that the connectome is necessary, but may not be the whole story. The roads predict the traffic patterns surprisingly well, but sometimes the same road carries different traffic, depending on the weather. It is still early. The complete zebrafish connectome has not been published, but when it is, it will be the first time we have a full wiring diagram of a vertebrate brain, a brain that is, in its fundamental architecture, much more similar to ours than any worm or insect. Vertebrate brains share a basic body plan, the same major regions: forebrain, midbrain, hindbrain, cerebellum, appear in fish, frogs, birds, mice, and humans. The zebrafish connectome will not be a human connectome, but it will be a connectome from our branch of the evolutionary tree, and that matters. When the complete zebrafish connectome arrives and researchers overlay the calcium imaging data onto it, when they can see every connection and every pattern of activity on those connections at the same time, we will have the first truly complete picture of a vertebrate brain in action. Not just the roads, not just the traffic. The roads and the traffic together, in one unified dataset. That will be the moment when the weight problem, at least for this organism, begins to have a quantitative answer. We will be able to measure precisely how much of the behavior the structure alone predicts, and how much lives in the dynamic, shifting, state-dependent activity that rides on top of it. That moment is close, perhaps very close, and whatever it reveals, it will reshape the entire conversation about what it means to copy a mind. There is something else the zebrafish has given us that deserves mention. Because the brain is so small and the imaging so complete, researchers can now perform what amounts to brain-wide correlation analysis. They can ask, when this neuron fires, which other neurons fire at the same time? Which neurons always fire together? Which ones never do? The result is a functional connectivity map. Not a structural one, like a connectome, but a map of coordinated activity. A picture of which neurons are in conversation with each other at any given moment. When these functional maps are compared to the emerging structural connectome, the overlap is significant, but not perfect. Neurons that are structurally connected tend to fire together, but some neurons that are not directly connected also show correlated activity, presumably communicating through intermediaries, or responding to the same neuromodulatory signals, or synchronized by oscillatory patterns that the structure alone does not predict. And some neurons that are structurally connected show little correlated activity, connections that exist physically but are functionally silent, like roads that are paved but carry no traffic. The zebrafish is the first place where we can see this gap between structural connectivity and functional connectivity in an entire vertebrate brain. And the gap is telling us something important. The map matters. The map matters a great deal, but the map is not the whole story. There is something riding on top of the map, something dynamic, something state-dependent, something that changes from moment to moment. And that something is part of what makes a brain a mind, rather than just a circuit. Breathe slowly. Let that accumulate. We are building a picture, and the picture is not simple. And then there is the mouse. The mouse brain has approximately 71 million neurons. That is roughly 500 times more than the fruit fly. A full mouse connectome does not exist yet, and it will not exist for several years. But pieces of it are being assembled, and the pieces are revealing. The Microns Project, a collaboration led by the Allen Institute for Brain Science, with partners at Princeton and Baylor College of Medicine, has mapped a cubic millimeter of mouse visual cortex at synaptic resolution. This is the largest volume of mammalian brain tissue that has been fully reconstructed. It contains roughly 100,000 neurons and hundreds of millions of synapses. But the Microns Project did something that no previous connectome effort had done at this scale. They recorded the activity of the same neurons before mapping their connections. They showed the mouse visual stimuli, movies, images, patterns, and recorded which neurons responded to which stimuli using calcium imaging. Then they preserved the tissue, sliced it, imaged it, and reconstructed the connectome. For the first time, scientists could ask, does the connectome predict the activity? If two neurons are connected, do they respond to similar things? If a group of neurons forms a tightly connected cluster, do they process the same type of visual information? Beat. The answer was yes. More connected neurons had more similar responses. Neurons that shared many synaptic connections tended to respond to the same visual features, the same orientations, the same directions of motion, the same spatial frequencies. The structure predicted the function, not perfectly, but significantly. The correlation was strong enough that you could, with moderate accuracy, predict what a neuron would respond to just by looking at what it was connected to. The roads, once again, predicted the traffic, but the prediction was not perfect. Some neurons that were strongly connected responded to quite different stimuli, and some neurons with weak structural connections showed highly correlated activity. The map was informative. The map was useful. The map was not the whole story, a theme that should be familiar by now. The FlyWire project, out of Sebastian Song's lab at Princeton, took a different approach. It crowdsourced the mapping of the mouse retina, enlisting more than 300,000 volunteers from around the world to trace neurons through electron micrograph stacks, turning connectome proofreading into something that looked almost like a video game. Volunteers logged into a web interface, saw a three-dimensional cube of neural tissue, and colored in the branches of a single neuron, extending it through the image stack one segment at a time. The interface was intuitive, almost addictive. People competed for accuracy. Leaderboards tracked the most prolific tracers. Some volunteers traced neurons for thousands of hours. What the Retina Project revealed was unexpected. The number of distinct cell types in the mouse retina, the number of functionally different kinds of neurons, each performing a specific computational role, was far greater than anyone had predicted. Textbooks had described perhaps a dozen types. FlyWire found more than 60. Each type had a distinctive shape, a distinctive pattern of connections, and a distinctive computational function. Some detected edges. Some detected motion in specific directions. Some responded only to changes in light level. Some fired only when a dark object moved against a bright background. An adaptation thought to help mice detect hawks overhead. The wiring was not simple. It was not a handful of circuit patterns repeated across the tissue. It was an elaborate, deeply structured architecture with dozens of cell types, each connected in specific ways to specific partners. And this was just the retina, a thin sheet of tissue at the back of the eye, not even the brain proper. The wiring was not simple. It was not a handful of circuit patterns repeated across the tissue. It was an elaborate, deeply structured architecture with dozens of cell types, each connected in specific ways to specific partners. Imagine you opened the hood of a car expecting to find a simple engine, and instead found a Swiss watch. Thousands of tiny components, each one precision, engineered, each one serving a specific function. The whole thing operating with a delicacy that makes the original expectation of simplicity seem naive. The more we map, the more complexity we find. Each rung of the ladder reveals not just more neurons, but more kinds of neurons. More kinds of connections, more layers of organization that were invisible at the previous scale. And this is the fundamental challenge of the enterprise. The territory is more complex than any map we have drawn so far, and every improvement in the resolution of our maps reveals new complexity that was hidden at the previous resolution. We are not converging on a complete understanding. We are discovering that the thing we are trying to understand is deeper than we thought. One projection from a company called AABio working on optical connectomics estimates that a complete mouse connectome could cost $30 million within 5 to 10 years. That sounds like a lot of money. It is not. The Human Genome Project cost roughly $3 billion in the 1990s. The H-Ligne dataset, 1 cubic millimeter of human brain, cost vastly more than $30 million in total research expenditure over its 10-year development. A complete mouse brain for $30 million would represent a cost reduction of several orders of magnitude from current methods. The cost is dropping. The methods are improving. The ladder is being climbed, rung by rung, organism by organism. But before we go further up, we need to go sideways, because there are two animals that challenge everything we think we know about where minds live. The common octopus has approximately 500 million neurons. That is more than a mouse, more than a rat, more than many mammals. But the octopus brain is structured in a way that no mammalian brain is. Roughly 2/3 of an octopus's neurons are not in its brain. They are in its arms. Each arm of an octopus contains a dense network of neurons, organized into ganglia, clusters of nerve cells that function as local processing centers. The arm ganglia can sense touch, taste, temperature, and pressure. They can coordinate complex movements. They can make what appear to be independent decisions. Let us describe what this actually looks like in practice. An octopus reaches into a crevice in a coral reef. The arm encounters an object. The neurons in the arm sense its texture, its shape, its chemical signature. The suckers that line the arm's underside contain chemoreceptors, molecular taste buds, that can distinguish food from rock from predator by touch alone. If the object is food, a crab, a clam, a shrimp, the arm grasps it, manipulates it, and passes it back toward the mouth. If the object is dangerous, a sea urchin, a stinging anemone, the arm withdraws. All of this can happen without any instruction from the central brain. The arm's local nervous system has enough processing power to perform the entire sequence. Detection, identification, decision, action on its own. We know this because of a striking experiment. If you sever an octopus arm from its body, the arm continues to respond to stimuli for hours. Not reflexively, not with simple withdrawal. The severed arm will reach for food. It will pull away from threats. It will curl and extend and explore its environment with purpose and coordination, even though it is no longer connected to anything that we would call a brain. This is not a reflex. A reflex is a simple circuit. Stimulus in, response out, no processing in between, a need. Jerk reflex involves 3 neurons. The severed octopus arm performs coordinated, goal-directed behavior involving thousands of neurons working together. It explores its environment. It responds differently to different kinds of stimuli. It makes choices. There is a deeper strangeness here. When an octopus moves, its central brain does not control each arm directly. It does not send detailed instructions. Extend this sucker, rotate this segment, grip with this much force. Instead, the brain sends high-level commands. Reach toward that object, bring it here. And the arm's local nervous system figures out the details. The arm translates the brain's intent into specific movements, using its own computational resources. This is a fundamentally different architecture from the one we are familiar with. In a human body, the brain controls movement in extraordinary detail. The motor cortex sends signals to specific muscles, adjusting force and timing with millisecond precision. The spinal cord handles some reflexes locally, but voluntary movement is centrally orchestrated. The octopus has something closer to a distributed computing system. The brain is the coordinator, not the micromanager. The arms are semi-autonomous processors. The mind, if we can call it that, is not located in one place. It is spread across a body. And octopuses are intelligent. Genuinely, remarkably intelligent. They solve puzzles. They learn by observation, watching another octopus open a jar and then doing it themselves. Consider what this requires. The observing octopus watches a demonstration through a transparent partition. It sees another octopus grasp a jar, rotate the lid with coordinated sucker movements, and extract the crab inside. Later, when given the same jar, the observer solves it on the first try, or nearly so, while naive octopuses that did not watch the demonstration take much longer or fail entirely. This is observational learning. It requires understanding the goal of another animal's actions and translating that understanding into one's own motor plan. Primates do this. Corvids do it. Very few other animals do. They use tools. Researchers have documented octopuses carrying coconut shell halves across the ocean floor. An awkward, stilted walk that makes them slower and more vulnerable to predators, and then assembling the shells into a shelter when they reach their destination. They climb inside, pull the shells together around them, and sit there, enclosed. This is not instinct. No octopus is born knowing what to do with a coconut shell. It is planning. It is delayed gratification. It is accepting a cost now. The awkward, slow, exposed walk. For a benefit later, the future shelter. They recognize individual human faces and behave differently toward people who have treated them well versus people who have annoyed them. In laboratory settings at the Seattle Aquarium and the University of Otago in New Zealand, researchers have documented octopuses that would squirt water at certain staff members and not others. The choice was consistent. It was not random. The octopus had categorized individual humans and assigned them different behavioral responses. One famous octopus at the Seattle Aquarium learned to unscrew the lid of its tank from the inside. Not the decorative lid, the maintenance lid. The one that humans use to access the tank for cleaning and feeding. The octopus figured out the threading, the direction you have to turn a screw top to loosen it, and applied coordinated force with multiple arms to unscrew the lid, while the aquarium staff were in another room. This happened more than once before the staff installed a latch. Another, at the University of Otago, squirted water at a light fixture it found annoying. The light was a bright overhead fluorescent positioned near its tank. The octopus aimed its siphon, the muscular tube it normally uses for jet propulsion, at the light and shot a focused stream of water at it, repeatedly, over the course of several days, until the water shorted out the electrical connection and the light went dark. The staff replaced the fixture. The octopus shorted it out again. This was not a random behavior that happened to have a convenient result. The octopus aimed. It aimed at the same target, repeatedly, across multiple days. It had a goal, and it pursued that goal with persistence. There is a behavior called play that is exceedingly rare in the animal kingdom. It requires doing something for no immediate survival benefit. No food, no mating, no shelter. Just doing something because it is. Apparently. Interesting? Researchers at the University of Tennessee documented octopuses in laboratory settings, repeatedly blowing jets of water at floating pill bottles, pushing them away, and then pulling them back over and over in what the researchers could only describe as play. The bottles had no food value, The octopuses were not hungry. They were just doing something with the bottles, interacting with them for what appeared to be the sheer experience of interacting. Octopuses in laboratories have been observed escaping their tanks, walking across the floor, entering neighboring tanks to eat the crabs inside, and then returning to their own tanks. They have been documented dismantling the plumbing of their aquarium systems. They have figured out how to open child-proof pill bottles. And perhaps most remarkably, they do all of this with a brain that is profoundly different from the brains of any other intelligent animal on Earth. Having diverged from the vertebrate lineage more than 500 million years ago, all of this intelligence, and it is intelligence, by any behavioral measure, emerges from a nervous system that is organized in a way that has no counterpart in the vertebrate world. What does this mean for the Connectome project? Beat. If you map the central brain of an octopus, just the brain, the part inside the head, you would have mapped only 1/3 of its nervous system. You would be missing the majority of its neurons and the majority of its computational capacity. A connectome of the octopus brain alone would be like mapping the roads in one city of a 3-city metropolitan area and claiming to understand the traffic of the whole region. But mapping the entire octopus, brain plus 8 arms, would be an order of magnitude more difficult. Each arm contains roughly 40 million neurons, organized in complex 3-dimensional ganglia. The connections between the arms and the central brain involve thick nerve bundles carrying signals in both directions. The system is not just distributed, it is dynamically coordinated, with information flowing up from the arms to the brain and back down again in continuous loops. The octopus suggests that the mind, or at least the neural substrate that produces intelligent behavior, does not have to live in one place. It can be distributed, spread across a body, decentralized in a way that challenges our intuition about where the self resides. And if the mind can be distributed across a body, what does that mean for uploading it? Would you need to map the entire octopus, brain, and all 8 arms to capture the animal's behavior? If you simulated only the brain, would the simulation sit there, confused and paralyzed, missing 2/3 of its computational substrate? Or would the brain simulation alone produce some diminished version of octopus behavior, a mind that knows what it wants to do but cannot figure out how? Beat. And if you did map the whole animal, all 500 million neurons across all 9 processing centers, would the resulting simulation behave like an octopus? Or would it behave like 8 separate creatures that happen to share a body? Is the unity of the octopus experience something that emerges from the physical connections between its distributed processes, or is it something else? Something about timing, about chemical signaling, about the shared fluid environment of a single body? Beat. These are not abstract questions. They are the actual engineering questions that anyone attempting to digitize a complex nervous system will eventually have to answer. And then there is the planarian. Breathe slowly. This one is going to challenge something fundamental. Everything we have discussed so far, the worm, the fly, the zebrafish, the mouse, and now the octopus, has been about where the mind lives and how the structure relates to function. The next animal on our ladder does not just challenge our understanding of how minds work. It challenges our understanding of where memories are stored. And the answer, if it holds up, is genuinely unsettling. Planaria are flatworms. They are small, simple, unremarkable, in almost every way. Most species are about 1 to 2 centimeters long. They live in freshwater, ponds, streams, the undersides of rocks. They eat dead things, mostly. They glide along surfaces on a layer of mucus, propelled by microscopic hairs called cilia. They have a brain, a tiny cluster of neurons in their head, connected to 2 nerve cords that run the length of their body. The brain contains roughly 10 to 20,000 neurons, depending on the species. They are not impressive animals. There is no reason, looking at them, to think they would have anything to teach us about the nature of the mind. And they can do something that no other animal with a brain can do. You can cut a planarian in half, and both halves will regenerate into complete worms. The tail half grows a new head. The head half grows a new tail. Within about 2 weeks, you have 2 complete, functioning worms where you had one. This is not unusual for simple animals. Many organisms can regenerate. Starfish can regrow arms. Salamanders can regrow limbs. Lizards can regrow tails. But planarian regeneration is different in a crucial way. The tail half, the half that had no brain, grows a new one. From scratch, it builds a new cluster of neurons, wires them together, connects them to the nerve cords, and within days the regenerated worm is navigating, feeding, and behaving normally. The new brain is not a copy of the old brain. It cannot be. The old brain was in the other half. The new brain is constructed from stem cells, pluripotent cells called neoblasts that exist throughout the planarian body and can become any cell type, including neurons. The worm does not regrow its brain from a backup copy. It builds a new one from raw materials, guided by the body's patterning signals. This alone is remarkable, but it is not the remarkable part. In 2013, a group of researchers at Tufts University, led by Michael Levin and Tal Shomrat, trained Planaria to find food in a specific location in a petri dish. The experimental setup was carefully designed: a circular arena with a rough floor and smooth walls, a food reward in a particular location. The worms had to learn to overcome their natural aversion to the open, illuminated center of the arena and navigate to the food source. The worms learned the task over repeated trials. Trained worms could reliably navigate to the food faster than untrained worms. They had formed an association between the environment and the reward, a simple memory. Nothing that would impress a dog or a crow, but a measurable, reproducible change in behavior as a result of experience. Then the researchers cut the trained worms in half and waited for them to regenerate. 2 weeks later, the regenerated worms, including the ones that grew new heads, new brains, entirely new nervous systems from the tail half, were tested on the same task. They appeared to remember the training. They found the food faster than untrained worms, not as fast as the originals, but faster than worms that had never been trained at all. The brain was destroyed, a new brain grew, and something about the training persisted. Let that sit for a moment. The organ that we believe stores memories was removed. A new organ grew in its place, and the memories, or something like them, some trace, some residue, survived the replacement. This result is contested. Not all labs have replicated it cleanly. The methodology has been debated. Some researchers argue that the faster performance could be explained by chemical cues lingering in the body, or by epigenetic changes in the neoblasts that biased the new brain's development, or by the regenerated worms simply being more active due to the stress of being cut in half. But the finding has been replicated, with variations, by multiple groups, and the Tufts lab has continued to publish follow-up studies with increasingly rigorous controls. The effect persists. Something transfers. Sit with this for a moment. An animal grows a new brain, and something that looks like memory survives. If this is real, and the evidence, though contested, continues to accumulate, it is one of the most profound findings in the history of neuroscience. It suggests that the brain is not the only place where experience leaves its mark, that the body itself carries some trace of what the animal has learned, that the connectome is necessary for the mind to function, but the information that shapes the connectome may live somewhere else entirely. The planarian is a mystery that sits at the heart of the entire connectome enterprise. It asks the question that the worm and the fly seem to answer so cleanly. Is the wiring diagram really where the mind lives, or is the mind something more distributed, more diffuse, more deeply embedded in the biology of the whole organism than any wiring diagram could capture? If memories can survive the complete replacement of the brain, then the connectome, at least in this organism, is not the complete story. Something else is carrying information, something outside the neurons, something in the body. Some researchers have proposed that memory might be stored in epigenetic modifications, chemical changes to DNA that alter gene expression without changing the genetic code. When a worm learns something, the theory goes, the experience modifies the epigenetic state of cells throughout the body, not just neurons. When the brain is destroyed and regrown, the new neurons inherit these epigenetic markers from the surrounding tissue, and the markers bias the new brain's development in ways that reconstruct something resembling the original memory. Others have suggested bioelectric patterns. Michael Levin's lab at Tufts has shown that Planaria maintain body-wide electrical gradients, voltage differences across their tissues that carry information about body shape and organization. These bioelectric patterns persist even when the brain is removed. They are maintained by gap junctions, tiny channels that connect neighboring cells and allow ions to flow between them. In theory, a bioelectric pattern could encode information that survives brain regeneration, providing a non-neural substrate for memory. We do not know which explanation, if any, is correct. The planarian remains a puzzle. But it is a puzzle that carries an important warning for the Connectome project. Mapping the brain may not be enough. The mind may extend beyond the brain, into the body, into chemical gradients and electrical patterns and biological substrates that no electron microscope can capture. The worm's connectome produced behavior in a Lego robot. The fly's connectome reproduced motor output at 95% accuracy. The octopus distributes its mind across its body. The planarian might store its memories outside its brain entirely. Each organism on the ladder tells us something different, and the message taken together is that the relationship between structure and mind is not simple. It is not a single answer. It is a landscape of possibilities, and we are only beginning to map its edges. The worm says yes. The map is the mind. The fly says mostly yes. The zebrafish says yes, but with caveats. The mouse says the complexity goes deeper than you think. The octopus says the mind might not live where you expect. The planarian says it might not live in the brain at all. No single organism tells the whole story. That is why we need them all. Each one illuminates a different corner of the question. And together, rung by rung, they are building the most detailed understanding of the mind that has ever existed. Before we move to the mechanics of mapping, to the microscopes and the data and the staggering math of scaling up, we need to sit with something for a while. The thing that makes this problem so hard is not the technology. The technology is improving every year. What makes this problem hard is that we do not fully understand what we are trying to capture. A connectome captures wiring. Every neuron, every synapse, every physical connection between cells. But a living brain is not just wiring. A living brain is wiring plus activity, plus chemistry, plus timing, plus 1,000 things that change from moment to moment in ways that a frozen map cannot record. Scientists who work on connectomics call this the weight problem. Not weight as in heaviness, weight as in the value assigned to each connection, the functional strength, the importance, the meaning of each synapse in the living, active brain. A connectome captures the topology, which neurons are connected to which. The weight problem asks, is topology enough? Let us break this down carefully, not quickly, slowly, piece by piece, because the details are the whole problem. Breathe slowly. There is no rush. The first piece of the weight problem is synaptic strength. Two neurons can be connected by a synapse. The synapse exists. It is physically there, and an electron microscope can photograph it. But the strength of that connection, how much signal actually passes through it when the first neuron fires, varies enormously, not by a little, by orders of magnitude. One synapse might reliably trigger the next neuron to fire every time. Another synapse, identical in appearance, might produce such a weak signal that the receiving neuron barely notices. To understand the range, picture two different telephone calls. In one, the signal is crystal clear, every word transmitted at full volume. The listener hears everything. In the other, the connection is barely there, a faint whisper on the line, the listener straining to hear anything at all. Both calls use the same telephone network. Both pass through the same wires, but the functional impact of the two calls is completely different. One changes the listener's day. The other is noise. That is the range of synaptic strength, same physical connection, vastly different functional meaning. How does this variation arise? Through experience, through use. A synapse that fires frequently gets stronger. The process is called long-term potentiation, abbreviated LTP, and it is one of the most well-studied phenomena in neuroscience. It was first described by Turgeir Lomo at the University of Oslo in 1966, and later characterized in detail by Tim Bliss and Lomo in a landmark 1973 paper published in the Journal of Physiology. When a synapse is used repeatedly, a cascade of molecular events unfolds. The sending neuron increases the number of synaptic vesicles, tiny packets of neurotransmitter, that it releases with each signal. The receiving neuron grows more receptors on its surface, becoming more sensitive to the neurotransmitter. Specifically, it inserts more AMPA receptors into its membrane, the receptors that mediate the fast, immediate response to glutamate, the brain's primary excitatory neurotransmitter. The physical structure of the synapse changes. It gets larger, more robust, more efficient. The dendritic spine on the receiving neuron swells. New proteins are synthesized and incorporated into the synapse. The connection is literally strengthened at the molecular level. The reverse also happens. A synapse that rarely fires gets weaker. This is called long-term depression. The receiving neuron pulls receptors off its surface. The dendritic spine shrinks. The synapse weakens. Eventually, if it remains unused for long enough, it may be pruned away entirely. The physical connection dissolved. The axon terminal retracted. The spine absorbed back into the dendrite, as if it had never existed. This is the cellular mechanism underlying learning and memory. When you learn a new skill, the synapses involved in that skill get stronger. When you forget something, the relevant synapses have weakened. Your memories are, in a very real sense, patterns of synaptic strength distributed across your brain. Every fact you know, every face you recognize, every skill you have practiced. Each one is encoded as a specific pattern of strong and weak synapses across specific circuits. This means that two connectomes that look identical, same neurons, same connections, same topology, could produce completely different behaviors if their synaptic strengths are different. The map is the same. The meaning of the map is different. Imagine two identical cities with identical road networks. Same streets, same intersections, same highways. But in one city, the highway between the suburbs and downtown carries 100,000 cars a day. In the other city, the same highway carries only a few hundred. The road exists in both cases, but the functional meaning of the connection is completely different. A road that carries a few hundred cars is an option. A road that carries 100,000 is the central artery of the city's economy. Can we measure synaptic strength from a connectome? Partially. Larger synapses tend to be stronger. The physical size of the synapse, the area of contact between the two neurons, correlates with functional strength. Electron microscopy can measure this, but the correlation is imperfect. Size is a proxy, not a measurement. Two synapses of the same size can have different numbers of receptors, different amounts of stored neurotransmitter, different molecular compositions that affect their function in ways that size alone does not capture. One might have recently undergone potentiation and be loaded with AMPA receptors. The other might be in the process of depression, its receptors being internalized even as its physical size has not yet changed. Same photograph, different function. Some researchers argue that size is good enough. that, for the purposes of simulation, the approximate synaptic strengths inferred from structural measurements are sufficient. The Fly simulation used structural proxies for synaptic strength, and it achieved 95% accuracy. Maybe close enough is close enough. Others argue that approximate is not the same as accurate, that the 5% gap in the Fly simulation and the much larger gaps that may emerge in more complex organisms are exactly where synaptic strength matters most. The rare, strong connection that drives a critical behavior. The recently weakened synapse that represents a recently extinguished memory. These details are lost in approximation. Let that settle. There is no rush. The second piece of the weight problem is neuromodulation. Your brain does not run on electricity alone. It runs on chemistry. And the chemical environment of the brain changes constantly in ways that are invisible to a connectome. Neurotransmitters like serotonin, dopamine, norepinephrine, and acetylcholine are released not just at synapses, but into broad regions of the brain, where they wash over millions of neurons simultaneously and change how those neurons respond to input. This is called volume transmission, as opposed to the targeted, point-to-point signaling of synaptic transmission. Imagine the difference between whispering a message to one person and shouting an announcement across a crowded room. Synaptic transmission is the whisper. Neuromodulation is the shout. Let us make this concrete. You are walking through a forest. It is quiet, dappled sunlight through the leaves, birds singing. Your brain is in a resting state. Moderate serotonin, low norepinephrine, tonic dopamine. You hear a sound, a branch snapping. In the fraction of a second, before you consciously register what happened, your brainstem floods your cortex with norepinephrine. Specifically, a tiny structure called the locus coeruleus, a cluster of only about 30,000 neurons in the brainstem, one of the smallest nuclei in the brain, sends projections to virtually every region of the cortex, releasing norepinephrine across your entire brain simultaneously. Your amygdala sends a burst of signals to the hypothalamus, which triggers the release of cortisol and adrenaline. In that moment, the same neurons with the same connections behave completely differently. Your sensory cortex becomes hyper-responsive. You hear sounds you would normally ignore. The signal-to-noise ratio in your auditory cortex shifts. Faint sounds that would have been filtered out are now amplified. Your prefrontal cortex narrows its focus. You stop thinking about dinner and start scanning for threats. Your motor cortex lowers its threshold. You are ready to run or fight on a hair trigger. Your hippocampus enters a heightened encoding state. Whatever happens in the next few minutes will be seared into long-term memory with unusual clarity. This is why people remember moments of fear in vivid detail years later. The wiring has not changed. Not one synapse has been added or removed. Not one connection has been strengthened or weakened. But the behavior of the entire system has shifted dramatically. The chemistry changed, and the chemistry changes the meaning of the wiring. This is neuromodulation, and it happens constantly. Not just in moments of fear. Every emotion you experience, every shift in attention, every transition from wakefulness to drowsiness, every fluctuation in mood, each one is accompanied by a change in the neuromodulatory environment. Dopamine surges when you anticipate a reward. Serotonin modulates your baseline mood. Acetylcholine sharpens your focus when you concentrate. Oxytocin floods your brain when you hold a newborn. Each one changes the functional meaning of every synapse it reaches. The same brain, the same connectome behaving in fundamentally different ways because the chemical context has shifted. This operates at a scale that a connectome cannot capture. A connectome is a structural snapshot frozen in a single moment. The chemical environment that gives the structure its meaning, the tide of modulators that rises and falls with every emotion, every state of alertness, every cycle of sleep and waking, is invisible to it. Consider the implications for copying a mind. Your connectome at 3 in the afternoon, after a cup of coffee in a brightly lit room, is structurally identical to your connectome at 3 in the morning, after a sleepless night in darkness. But the brain those two connectomes produce is profoundly different. One is alert, focused, capable. The other is foggy, distractible, slow, same roads, completely different traffic. And the difference is not in the roads. It is in the weather. Some researchers have tried to address this by mapping the distribution of neuromodulatory receptors, identifying which neurons have receptors for dopamine, which have receptors for serotonin, and so on. This adds a layer of information to the connectome, but it is a static layer. It tells you which neurons can respond to dopamine. It does not tell you how much dopamine is present right now, or how fast the level is changing, or what triggered the release in the first place. The weather analogy is apt. A connectome with receptor maps is like a road map that also shows which roads have drainage. You know which roads will be affected by rain. You do not know whether it is raining. Breathe slowly. We are in the middle of the weight problem. Let the pieces accumulate. The third piece of the weight problem is timing. Neurons do not simply fire or not fire, like switches that are on or off. They fire in patterns, bursts and pauses, rhythms and syncopations. The precise timing of when a neuron fires relative to its neighbors carries information. This is called temporal coding, and it is one of the most important concepts in modern neuroscience. Two neurons firing simultaneously mean something different than the same two neurons firing 50 milliseconds apart. A neuron that fires at 40 Hz carries different information than the same neuron firing at 80 Hz, even though the wiring is identical in both cases. To make this tangible, think about music. Two musicians can play the same notes on the same instruments in the same room, same equipment, same score. But if one plays the notes in rhythm and the other plays them out of time, the same notes, just shifted by fractions of a second, the result is completely different. One is a song, the other is noise. The notes are the structure, the timing is the meaning. Neurons work the same way. The information they carry is encoded, not just in which neurons fire, but in when they fire relative to each other, down to the millisecond. This is sometimes called spike timing, and it adds a dimension of information that is entirely invisible to a structural map. Some of these timing patterns are stable and predictable from structure. The visual cortex processes information in a sequence. Light hits the retina. Signals travel through the thalamus to the primary visual cortex, then to secondary areas, then to association areas. This sequence is determined by the wiring. The connectome predicts it, but other timing patterns are emergent and unpredictable, oscillations, rhythmic patterns of firing that sweep across large regions of the brain, are generated by networks of neurons interacting with each other. The gamma rhythm, for instance, oscillates at 30 to 100 Hz and is associated with attention and conscious awareness. It is produced by the interplay of excitatory and inhibitory neurons in local circuits, specifically by a dance between pyramidal cells and fast, spiking into neurons that take turns exciting and inhibiting each other in a rapid alternating pattern. The connectome describes the circuits, but whether the circuits produce gamma oscillations or some other pattern depends on the current state of every neuron in the circuit, which depends on what happened in the previous millisecond, which depends on neuromodulation, which depends on the organism's state and history. There is a phenomenon called spike timing dependent plasticity, SDP, that makes timing even more fundamental. If neuron A fires just before neuron B, the synapse from A to B gets stronger. If A fires just after B, the synapse gets weaker. The window is narrow, roughly 20 to 40 milliseconds, but within that window, the direction of the timing difference determines whether a connection is strengthened or weakened. This means that timing does not just carry information. Timing physically reshapes the connectome itself. The activity changes the map. The traffic redraws the roads. Consider what this means for a connectome-based simulation. You have mapped every neuron and every connection. You run the simulation. But at what timing? With what initial conditions? The same network, started in two slightly different states, can produce wildly different patterns of activity within seconds. A phenomenon familiar from chaos theory, where small differences in starting conditions lead to large differences in outcomes. The brain is not a clock that always ticks the same way. It is a system poised on the edge of many possible states, and the timing of its activity is what selects among them. These temporal patterns change constantly. They encode everything from the pitch of a sound to the position of your hand to the feeling of surprise. They are the conversation happening on the wires, and the connectome only shows you the wires. Breathe slowly. 2 more pieces remain. The fourth piece of the weight problem is the cells we used to ignore. For decades, neuroscience focused almost exclusively on neurons. They fire, they transmit signals, they do the obvious computational work. But the brain contains roughly as many glial cells as neurons. Some estimates put the ratio at nearly 1:1 in the human brain. Roughly 85 billion glial cells alongside 86 billion neurons. And a specific type called astrocytes turn out to be far more than the structural scaffolding they were once believed to be. The name astrocyte means star cell because of their shape, a central body with branching processes that extend in all directions like the points of a star. For a long time, scientists thought astrocytes just held the brain together. Structural support, metabolic housekeeping, cleaning up waste, important but not interesting. The very name glia comes from the Greek word for glue. That was the prevailing view for more than a century. Then, starting in the 1990s, a series of discoveries changed the picture. In 1990, Anne Cornell, Bell and colleagues at Yale published a paper showing that astrocytes in culture could transmit calcium waves from one cell to another, a form of communication that did not involve electrical signaling at all. The waves traveled slowly, over seconds rather than milliseconds, but they traveled. The glue was talking. Astrocytes enwrap synapses. Their processes extend to surround the junction between 2 neurons, forming what is called the tripartite synapse. Not 2 elements, but 3. The sending neuron, the receiving neuron, and the astrocyte. A single astrocyte can enwrap thousands of synapses simultaneously. Estimates suggest that a single human astrocyte contacts between 300,000 and 2 million synapses. Picture an octopus. There is that animal again, with thousands of arms, each arm wrapped around a different conversation between neurons, listening to all of them at once. Astrocytes sense when neurons fire. They detect the neurotransmitter released at the synapse. In response, they release their own chemical signals, called glia transmitters, that modulate synaptic transmission. They can make a synapse stronger or weaker. They can change the probability that a signal will be transmitted. They can alter the timing of neural responses. They regulate the concentration of neurotransmitter in the synaptic cleft, controlling how long a signal lasts. They uptake excess glutamate, the brain's primary excitatory neurotransmitter, preventing it from accumulating and overstimulating neurons to death, a process called excitotoxicity. And astrocytes form networks of their own. They are connected by gap junctions, physical channels between adjacent cells that allow ions and small molecules to flow freely from one astrocyte to another. Through these gap junctions, astrocytes can pass signals across vast distances within the brain, coordinating activity across regions that are far apart in the connectome. An astrocyte in one cortical layer can communicate with an astrocyte in another layer, or even another brain region, through a chain of gap junction connected cells. A kind of slow chemical telephone network running alongside the fast electrical network of neurons. The astrocyte network operates on a different timescale than the neural network. Neurons fire in milliseconds. Astrocytes respond over seconds to minutes. They integrate information over longer windows, smoothing out the rapid fluctuations of neural activity and providing a slower, more sustained form of modulation. If neurons are the rapid-fire telegraph operators of the brain, astrocytes are the postal service, slower, but covering far more ground with each delivery and carrying messages that shape the context in which the telegraph operates. A connectome that maps only neurons is mapping half the system. The other half, the glial network, the astrocytic web, is a parallel communication system that we are only beginning to understand, and no one has ever mapped it. The FlyWire connectome does not include glial cells. The HO1 dataset identifies some astrocytes, but does not reconstruct their networks. We are building maps of a city's roads, while ignoring its subway system. And there is growing evidence that astrocytes may be involved in higher cognitive functions, not just housekeeping. Studies in mice have shown that disrupting astrocyte calcium signaling impairs learning and memory. Other studies have found that astrocyte networks synchronize across brain regions during sleep, suggesting a role in memory consolidation. The glue, it turns out, may be doing some of the thinking, or at least shaping the conditions under which thinking happens. Breathe slowly. And finally, the fifth piece of the weight problem, the body. The planarian reminds us. The octopus reminds us. The brain does not exist in isolation. It is embedded in a body, connected to sensory organs and muscles and glands, and an immune system and a gut microbiome that communicates with the brain through the vagus nerve. The gut-brain axis is one of the most active areas of research in neuroscience right now. Your gut contains roughly 500 million neurons, more than the octopus has in its entire central brain. These neurons form the enteric nervous system, sometimes called the second brain. They regulate digestion, of course, but they also communicate with the brain through the vagus nerve, sending signals that influence mood, appetite, anxiety, and even social behavior. The vagus nerve is the longest cranial nerve in the body. It wanders. The word vagus comes from the Latin for wandering, from the brainstem to the abdomen, branching along the way to connect to the heart, the lungs, the liver, the stomach, and the intestines. It carries signals in both directions. The brain sends instructions down. The gut sends information up, and the information flowing up about the state of digestion, the composition of the gut microbiome, the presence of inflammation, influences brain function in ways that are only now being quantified. The gut microbiome, the trillions of bacteria living in your intestines, produces neurotransmitters, actual neurotransmitters, serotonin, dopamine, GABA, roughly 95% of the serotonin in your body is produced in your gut, not in your brain. Changes to the gut microbiome have been shown to affect behavior in animal studies, altering anxiety levels, social behavior, and even cognitive performance. Germ-free mice. Mice raised in completely sterile environments with no gut bacteria show altered stress responses, abnormal social behavior, and impaired memory compared to mice with normal microbiomes. Transplant gut bacteria from anxious mice into calm mice, and the calm mice become more anxious. The bacteria are, in some measurable way, contributing to the animal's mental state. This does not mean your gut bacteria are thinking for you, but it means the brain is not operating in isolation. It is receiving a constant stream of chemical signals from the body, from the gut, from the immune system, from the endocrine system, from the peripheral nervous system, and these signals shape its activity. The brain is not a computer sitting on a desk. It is an organ inside an animal, and the animal's body is part of the computation. Remove the brain from the body, and you have not extracted the mind. You have amputated it from the context that gives it meaning. Think about what this implies for the project of copying a mind. If you scanned someone's brain and simulated it perfectly, every neuron, every synapse, every connection, every synaptic weight, but ran the simulation without a body, without a gut, without a heart sending rhythmic pulses of blood pressure that the brain uses to calibrate its sense of time, without the constant low hum of proprioceptive signals from muscles and joints that tell the brain where it is in space, what would happen? Would the simulation feel like a person floating in darkness? Or would it feel like nothing at all? A mind so deprived of the inputs it was built to expect that it cannot function. A fish on a table, gasping, not because the fish is broken, because the water is gone. We do not know. No one knows. But the body problem reminds us that the question is not just about copying the brain. It may be about copying the entire context in which the brain operates. The body, the senses, the chemical tides, the gut, the beating heart. So here is the weight problem, stated plainly. A connectome is a photograph of a city's road network. Every road, every intersection, every bridge, every tunnel. The photograph tells you where traffic can flow. It does not tell you where traffic is flowing right now. It does not tell you how fast, or which direction, or whether there are accidents blocking 3 lanes. It does not tell you about the weather, or the holidays, or the construction, or the million individual decisions that each driver makes based on their own destination and their own history. And it does not tell you that the city's character changes completely between 3 in the morning and 5 in the afternoon, even though the roads are exactly the same. 5 pieces: synaptic strength, neuromodulation, timing, glia, the body. Each one a layer of information that a structural connectome does not capture. Each one potentially necessary for understanding what the brain does, as opposed to merely what the brain is. The map is necessary. No one disputes that. Without the roads, there is no traffic. Without the connectome, there is no neural activity. The structure is the foundation on which everything else rests. But the map may not be sufficient. The traffic, the dynamic, flowing, constantly changing pattern of activity that rides on the structure may be where the mind actually lives. And yet, the fly simulation achieved 95% accuracy from the map alone. This is the tension. The theory says the map should not be enough. The data says it might be. The resolution may come as we climb higher on the ladder, to organisms complex enough that the gap between structure and behavior becomes visible, or to organisms where the gap does not appear at all. We do not know yet. We are still climbing. Breathe slowly. Let the 5 pieces settle: synaptic strength, neuromodulation, timing, glia, the body. 5 layers between the map and the mind. 5 reasons the connectome might not be enough. And 1 stubborn data point: 95% accuracy in the fly. That says it might be. The tension is real, and it is unresolved, and that is where the science actually is. The hardest part is behind us. Let us talk about the machines. To map a brain, you have to destroy it. Let that sentence sit for a moment. Read it again if you need to. It is the single most important fact about the current state of connectome science, and everything that follows. The technology, the cost, the timeline. The ethical questions we will explore next time flows from it. This is not a metaphor. It is the literal physical reality of connectome mapping at the resolution required to see individual synapses. The process of reading the brain erases the brain. Every slice you image is gone. Every layer you ablate is consumed. You are reading a book by burning each page after you photograph it. The primary method is called serial section electron microscopy. Here is how it works. You take a piece of brain tissue. It must be preserved first. We will discuss that shortly. You fix it chemically, typically with glutaraldehyde and osmium tetroxide, which link proteins and stain lipid membranes, so they are visible under an electron beam. You embed the fixed tissue in resin, hard, transparent, stable, so it can be sliced cleanly. Then you begin to slice. Each slice is between 30 and 50 nanometers thick. For comparison, a single red blood cell is about 7,000 nanometers wide. A virus is about 100 nanometers. These slices are in the range of large molecules. You are cutting tissue at a scale where individual protein complexes are visible. You place each slice on a small grid, or tape. You feed it into a transmission electron microscope and photograph it at a resolution of 4 to 8 nanometers, fine enough to see individual synaptic vesicles, the tiny bubbles of neurotransmitter that carry signals across the synaptic gap, fine enough to distinguish excitatory synapses from inhibitory ones, based on the shape and density of the post. Synaptic density, the thickened region of the receiving neuron's membrane at the synapse. Then you move to the next slice, and the next, and the next. The tissue you just photographed cannot be re-imaged. The electron beam damages it. The slice cannot be restacked. The destruction is the method. There is a newer technique called FibSEM, focused ion beam, scanning, electron microscopy. Instead of cutting with a physical blade, a beam of gallium ions ablates the surface of the tissue 1 nanometer, thin layer at a time. Each newly exposed surface is immediately imaged by a scanning electron microscope pointed at the block face. The resolution is isotropic, equal in all 3 dimensions, at 4 to 8 nanometers. This makes reconstruction easier, because the images are evenly spaced in all directions. FibSEM is slower per volt volume than serial section M, but it is more automated. There are no physical sections to handle, no ribbons of tissue to collect and mount. The machine runs continuously, ablating and imaging, ablating and imaging. The FlyWire brain was imaged using a combination of these techniques. A third approach, still emerging, is called expansion microscopy. Instead of slicing the tissue thinner, you make the tissue bigger. You embed it in a gel that swells when you add water, physically expanding the tissue by a factor of 4 to 10 in each dimension. A structure that was 100 nanometers across becomes 400 to 1,000 nanometers, large enough to see with a conventional light microscope instead of an electron microscope. Light microscopes are faster, cheaper, and more widely available than electron microscopes. If expansion microscopy can achieve the resolution needed for connectome mapping, and recent papers suggest it is getting close, the cost and speed of mapping could improve by orders of magnitude. The technique was invented by Ed Boyden's group at MIT in 2015, and it has been refined rapidly since then. The most recent versions achieve effective resolutions, approaching those of electron microscopy, at a fraction of the cost and time. It is one of the most promising developments in the field. Each of these approaches, serial section, M, FibSEM, expansion microscopy, has its own strengths and limitations. No single method is optimal for all purposes. The future of connectomics will likely use all of them, choosing the right tool for each scale and each question. Now, let us talk about scale. In 2024, a team from Google and Harvard published the HO1 dataset. It represents 1 cubic millimeter of human cerebral cortex, a piece of brain tissue roughly the size of a grain of sand, from a surgical epilepsy patient, tissue that was already being removed as part of treatment. This grain of sand was sliced, stained, imaged, and reconstructed over approximately 10 years. The result, 57,000 cells identified, 150 million synapses catalogued, 1.4 petabytes of raw data. That is 1.4 million gigabytes. For a single cubic millimeter. To appreciate the scale of the reconstruction, consider this. The raw images form a 3-dimensional volume. If you printed every image at standard resolution and stacked them, the stack would be taller than a 10-story building. The data, stored on hard drives, would fill a room, and this represents a volume of brain tissue smaller than a sesame seed. The paper was published in Science, You can browse the dataset yourself online, zoom into individual neurons, trace their axons and dendrites, see the synapses connecting them. It is the most detailed portrait of neural tissue ever created. And even in this tiny volume, the researchers discovered things that had never been seen before. They found neurons with unusual morphologies, shapes and branching patterns that did not match any known cell type. They found axons that wrapped around the cell bodies of other neurons in tight spirals, a wiring pattern that had never been described in any species. They found that the density and organization of synapses varied dramatically across even this small patch of cortex, suggesting a level of structural heterogeneity that textbooks had not predicted. 1 cubic millimeter, and it was full of surprises. Every time a new volume of brain tissue is mapped at this resolution, the reaction among neuroscientists is the same. We did not expect this. The brain keeps revealing layers of organization that were invisible at coarser scales. It is like mapping the ocean floor. Every time the sonar gets more precise, new mountains and canyons appear that no one predicted. The territory is more complex than any model anticipated, and the territory we have mapped is vanishingly small compared to what remains. Let that settle. 1 cubic millimeter, 1/1,000,000th of the whole. And already, within that fragment, structures no one had seen before, patterns no textbook described. Imagine what waits in the other 1,259,990, 9 cubic millimeters. And now, the arithmetic. The human brain is approximately 1,260,000 cubic millimeters. The HO1 dataset mapped one of them. To map the entire brain at the same resolution would require roughly 2 exabytes of raw data. An exabyte is 1 billion gigabytes. 2 exabytes is more data than the entire Library of Congress has produced in its history, multiplied by several thousand. The scanning time on a single microscope running continuously would be 500 to 5,000 years, depending on the technique. Think about that. At the slow end, you would need to start scanning today and continue without interruption until the year 2526. The pyramids of Giza would be closer to us in time than the completion of the scan, with 100 parallel systems, machines running in multiple laboratories around the world, each one imaging a different section of brain continuously. It would still take 5 to 50 years, but the scanning is not the real bottleneck. The real bottleneck is the proofreading. The raw images must be segmented, each neuron traced through thousands of slices, each synapse identified and classified. AI does most of this work now. The algorithms have improved dramatically since the FlyWire project. But the AI still makes errors. And in a structure as complex as the human brain, even a small error rate produces millions of mistakes. At current methods, a complete human connectome would take 50 to 100 years and cost between $100 billion and $600 billion for one brain. But the cost is changing, and it is changing fast. Emerging methods are attacking the problem from multiple angles. Optical connectomics, using expansion microscopy and high-speed light microscopes, could reduce imaging time by 1 to 2 orders of magnitude. Sequencing-based approaches that skip electron microscopy entirely. Techniques like BARSEC and MAPSEC that use viral barcodes to trace connections between neurons could potentially map connectivity patterns without imaging at all. AI-guided adaptive imaging, where the algorithm decides in real time which regions need higher resolution and which can be scanned more quickly, could reduce the total data volume by an order of magnitude. eBio estimates a complete mouse connectome at $30 million within 5 to 10 years. A complete human connectome, using next-generation methods, might cost in the low billions rather than hundreds of billions. Still enormous. But within the range of large-scale government science projects, these numbers are not arguments against the project. They are the honest shape of the problem and the shape is changing faster than most people realize. The Human Genome Project was declared impossible by serious scientists at its outset. It was completed ahead of schedule. The cost of sequencing a human genome dropped from $3 billion to roughly $200 in 2 decades. Connectomics may follow a similar curve, or it may not. We cannot know in advance, but the trajectory so far points steeply down. While one group of scientists works to map the brain by destroying it, another group is building devices that read the brain while it is still alive. These are not the same project, but they are approaching the same frontier from opposite directions, and the distance between them is closing. In 2024, Neuralink implanted a brain-computer interface in a human patient for the first time. Noland Arbor, a young man paralyzed from the neck down, received an implant called the N1, approximately 1,020, 4 electrodes on 64 ultra-thin threads, inserted into the surface of his motor cortex by a surgical robot. Through the device, Arbor can move a cursor on a screen by thinking about moving his hand. He can type. He can browse the internet. He can play video games. He played chess online within weeks of the implant. He played Civilization. He browsed social media. He described the experience as intuitive, like using the Force, in his words. After years of complete paralysis, he can interact with a computer using nothing but his thoughts. This is extraordinary. It is life-changing for Arbor and the patients who will follow him. It is a genuine achievement of engineering. It is also, by the standards of connectome mapping, almost unimaginably crude. Neuralink's 1,020. 4 electrodes read the aggregate activity of a small patch of motor cortex. Each electrode picks up signals from roughly a handful of nearby neurons, maybe 10 to 50. The device reads somewhere between 1,000 and 50,000 neurons, depending on how you count. Out of 86 billion, the resolution differs from connectome, mapping by 6 orders of magnitude. Neuralink knows that a group of neurons in this general area fired. The connectome knows which specific neuron connected to which other specific neuron through which specific synapse. The difference is like the difference between knowing that somewhere in Chicago someone is making a phone call and knowing who called whom on what phone, at what time, and what they said. But resolution is increasing. Neuralink's next-generation devices aim for 4,096 electrodes. Academic labs are developing probes with tens of thousands of recording sites. The density of electrodes is doubling roughly every few years, a kind of Moore's law for neural recording. A company called Control Labs, acquired by Meta in 2019 for a reported $500 million to $1 billion, built a wristband that reads motor neuron signals from the forearm. No surgery, no implant. You wear it on your wrist, and it decodes your intended hand movements from electrical patterns in the muscles and nerves of your arm. The approach is fundamentally different from Neuralink. Instead of recording from the brain directly, Connecticut RL Labs intercepts the signals as they travel down the arm. The motor cortex generates an intent. Move this finger. Grip this object. And that intent is translated into patterns of nerve activation that travel through the arm. The wristband reads those patterns and decodes them. The resolution is lower than Neuralink. You cannot decode complex thoughts from arm muscles, but you can decode fine motor control with impressive precision, typing without a keyboard, controlling a virtual hand with individual finger movements, subtle gestures that could replace a mouse or trackpad, and BrainGate, an academic consortium spanning Brown University, Stanford, Case Western Reserve, and Massachusetts General Hospital, has been running clinical trials in paralyzed patients for longer than Neuralink has existed as a company. Their Utah array implant has 96 to 128 electrodes, fewer than Neuralink, but peer-reviewed, replicated, published in the highest impact journals, and tested in patients over periods of years. BrainGate patients have used their implants to control robotic arms, to type at rates of up to 90 characters per minute by imagining handwriting, and to communicate for the first time in years. The research has produced some of the most rigorous neuroscience of the decade. These technologies are not the same project as connectomics. The resolution gap is vast. Neural interfaces read the living brain at the scale of hundreds or thousands of neurons. Connectomics maps the structural brain at the scale of every individual synapse. They are looking at the same thing from opposite ends of a very long telescope, but both are improving. Neural interfaces are getting more electrodes, smaller probes, better algorithms. Connectomics is getting faster microscopes, better AI, cheaper processing, somewhere between them, at a resolution no one has yet achieved. The two approaches converge. A device that can read the living brain at synaptic resolution, a map that can be made without destroying the tissue. We do not know when. We do not know if it is 10 years away or 100, but the gap is closing from both sides. And when it closes, the question we started with tonight, can you copy a mind, will stop being theoretical. It will become an engineering question, a question of cost and time and resolution and political will. And those questions have a very different character than theoretical ones. Theoretical questions can remain unanswered for centuries. Engineering questions, once the physics permits them, tend to get solved within decades. Breathe slowly. We are almost at the end now. Let us take a moment to see where we are. We have been climbing a ladder. Each rung an organism. Each organism a lesson. We mapped the complete connectome of the simplest animal with a nervous system, 302 neurons, and the structure alone produced behavior. A Lego robot running the worm's wiring diagram moved like a worm. No programming, no instructions. The map was the mind. We mapped the complete connectome of an insect brain, 139,000 neurons. And the structure alone reproduced motor behavior at 95% accuracy. The wiring diagram, simulated on a computer, produced a virtual fly that moved almost identically to a real one. We watched every neuron in a zebrafish brain firing simultaneously, and we saw that the structure predicts the activity, but the activity also depends on the animal's internal state. The roads predict the traffic, but the weather changes the traffic too. We mapped pieces of the mouse brain at synaptic resolution and found that connected neurons respond to similar stimuli. Structure predicts function, and we found more than 60 types of neurons in the retina alone, where textbooks had described a dozen. Complexity increases faster than our categories can capture. We discovered that the octopus distributes its mind across its body. 2/3 of its neurons are in its arms. Sever an arm and it keeps behaving. The mind does not have to live in one place. We discovered that the planarian may store memories outside its brain entirely. Cut off the head, grow a new one, and the memories persist. The connectome, at least in this organism, is not the complete story. We measured the weight problem, the gap between the map and the traffic, and found 5 layers of information that a structural connectome does not capture: synaptic strength, neuromodulation, timing, glial networks, and the body, each one potentially necessary for a complete picture of the mind. And we scanned 1 cubic millimeter of human cortex, the most detailed portrait of neural tissue ever made. 57,000 cells, 150 million synapses, 1.4 petabytes of data, the size of a sesame seed, 1/1,000,000th of the brain. None of this existed 30 years ago. Most of it did not exist 10 years ago. Some of it did not exist 2 years ago. The pace is accelerating. Each advance enables the next. Better microscopes produce more data. More data trains better AI. Better AI enables faster proofreading. Faster proofreading makes larger connectomes feasible. Larger connectomes reveal new principles. New principles guide the next experiment. The wheel turns, and it turns faster with each revolution. There is no rush, but there is momentum, and the momentum is real. And here is what remains. Scale: 1.26 million times more tissue to map than the largest human dataset we have. Cost: $100 to $600 billion at current methods. But the methods are changing time, 50 to 100 years, unless the methods change dramatically, and the methods are changing dramatically. The weight problem, open narrowing, not yet resolved. The fly says structure might be enough. The planarian says it might not. The zebrafish says it depends on context. We need more data. The preservation problem: you cannot map what you cannot fix and freeze. The scanning destroys the tissue. If you want to map a brain, you need to preserve it first, at nanometer fidelity, without ice crystals, without chemical artifacts, without any form of degradation that would blur the connectome into noise. And that preservation technology exists. It works. It has won prizes. But it raises questions of its own. Questions about identity, about death, about what it means to preserve a mind that is no longer running. We will explore those questions next time. The preservation problem: how you fix and freeze a brain at the moment of death so that its connectome remains intact. The copy problem: whether a perfect replica of your brain is you, or merely something that acts like you. The philosophy that no amount of data can resolve. What it would actually mean to be the first digitized mind. Whether the copy wakes up and remembers breakfast. Whether it matters if it does. Tonight, we stay here, on the ladder, looking up at a destination we can describe in extraordinary detail, but cannot yet reach. The first digitized mind, if it comes, will not arrive with a press conference. It will not feel like a moment. There will be no switch that someone throws, no screen that flickers to life showing a face that says hello. It will be a threshold crossed gradually, over years, in increments so small that no single paper marks the boundary. A slightly better simulation, a slightly more complete map, a slightly more accurate reproduction of behavior. And then, at some point, someone will run a simulation, and the result will be indistinguishable from the original, and the scientists will look at each other and realize that the line was crossed sometime last Tuesday, and no one noticed. It will be a threshold crossed quietly, in a laboratory, by people who spent their careers reducing a problem that once seemed infinite to one that merely seems very, very large. The worm taught us that structure can produce behavior. The fly taught us that this principle scales. The octopus taught us that minds can be distributed. The planarian taught us that memories might live outside the brain. The zebrafish is showing us the structure and the activity at the same time. The mouse is teaching us that the complexity goes deeper than we thought, and the single cubic millimeter of human cortex is reminding us, with every surprise it contains, that the brain is the most complex object in the known universe, and we have barely begun to read it. The science is patient. It does not care about our timelines or our impatience. It moves at the speed of evidence, and the evidence is accumulating faster now than at any point in history. Every year the microscopes get faster, the AI gets better, the maps get larger, the simulations get more accurate, the organisms on the ladder get more complex. And the question, at the top of the ladder, can you copy a mind, and if you do, is it still you, gets closer to being answerable. Not answered, closer to answerable. And that distinction, too, is important. The question it is building toward has no clean answer yet. That is not a failure. That is where the frontier actually is, the place where the known ends and the unknown begins. The place where the data runs out and the questions take over. The place where the smartest people in the world look at what they have built and what they have measured, and they say, honestly, we do not know yet. But we know more than we did yesterday. And tomorrow, we will know more than we do today. That is how science works. Not in leaps, in steps. Patient, careful, accumulated steps. Each one a rung on the ladder. Each one bringing the question into slightly sharper focus. Each one making the impossible slightly less impossible. each one honoring the tradition that Sidney Brenner began in a laboratory in the 1970s. When he sat down in front of a stack of electron micrographs and began to trace, by hand, the 302 neurons of a worm that no one had ever bothered to look at closely before, he could not have known where that work would lead. He could not have imagined flies and zebrafish and cubic millimeters of human cortex and robots made of Lego bricks that move like worms. He just knew the question was worth asking, and so he began. Rest now. The ideas are patient, and so is the science.